Welcome to our video about the fascinating world of Pokemon and their placement on the Tree of Life. Today, we take a closer look at Mook and its relationship with all other Pokemon as they are all part of the Archaea clade. Ready? Let's go! Archaea, formerly also known as Archaebacteria, Archaebacteria, or primitive bacteria, form one of the three domains into which all cellular organisms are classified. The other two domains are bacteria, which together with Archaea are grouped as prokaryotes, and eukaryotes, eukaryota, which, unlike prokaryotes, possess a nucleus enclosed by a nuclear membrane. Archaea, however, have closed DNA molecules, circular chromosomes, like bacteria, that exist in the cytoplasm as a nucleoid without a membrane. Archaea are single-celled organisms. So far, no pathogenic archaea have been identified. In many molecular biological characteristics, archaea are more similar to eukaryotes than to bacteria. Nevertheless, they exhibit typical bacterial traits, such as cell size, the absence of a nucleus, the mode of cell division, the presence of a closed DNA molecule, relatively simple motility structures, flagella, and, like bacteria, ribosomes with a sedimentation coefficient of 70s, although archaeal ribosomes have a more complex structure. The genes of both domains are organized in so-called operons. Archaea can also carry plasmids, as seen in an Archaean, provisionally named Sulfolobus NOB8H2, from the Sulfolobaceae family in the Crenarchaeota phylum. However, the central molecular processes, such as translation and transcription, are quite similar to those in eukaryotes. Archaea use RNA polymerases composed of multiple protein subunits, which are resistant to rifampicin and streptolitogen. Translation involves initiation and elongation factors that are very similar to those in eukaryotes, and transcription starts at a TATA box. Archaea also possess many unique characteristics, especially in their cell wall structure, which differ significantly from the other domains. Archaeal cell walls contain pseudopeptidoglycan, pseudomurian, and display great diversity in their composition. Some archaea lack a cell wall entirely, thermoplasma, while others have highly complex, multilayered cell walls, methanosporellum. Due to their structural differences, archaea are generally resistant to cell wall targeting antibiotics. Additionally, the composition of the archaeal plasma membrane differs. In bacteria and eukaryotes, fatty acids are bound to glycerol molecules via ester bonds, whereas in archaea, glycerol diethers or even bisglycerol tetraethers, monolayer membranes, are present, along with branched isoprene chains instead of simple fatty acids. Hyperthermophilic archaea often have highly stabilized cell membranes, glycerol tetraether, which not only provide enhanced thermal stability but also serve as adaptations to acidic environments. Some archaeal species can move remarkably fast relative to their size. With speeds of 400 to 500 bodies per second, (bps), Methanocaldococcus janaskii and Methanocaldococcus velocis, both from the genus Methanocaldococcus, are the fastest organisms ever measured. For comparison, a sports car traveling at 400 bps would reach speeds of over 6,000 km per hour, close to hypersonic velocity. In contrast, the bacterium Escherichia coli moves at around 20 bps, similar to a cheetah. Many cultivated archaeal species thrive in extreme environmental conditions. Some grow best at temperatures above 80 degrees Celsius, hyperthermophilic, others live in highly concentrated salt solutions, halophilic, while some inhabit strongly acidic environments, with pH levels even below zero. Acidophilic, or highly alkaline environments, pH above 10, alkalophilic. Members of the genus Pacrophilus, P. oshimi and P. torridus, order Thermoplasmatales, have an optimal growth pH of 0.7 and can even survive at -0.06 pH. Archaea are of interest in research because they may preserve traits from early life on Earth. Their extraordinary metabolism is also notable. For example, some can grow at 110 degrees Celsius. Most known archaeal species are autotrophic, meaning they do not require organic compounds for growth but instead assimilate carbon dioxide to build their cellular structures. However, heterotrophy, where carbon is obtained from organic compounds, is also widespread. Most cultivated archaea exhibit anaerobic metabolism, and many anaerobic archaea are toxic to oxygen, O2. One unique metabolic feature of Archaea is methanogenesis, a process performed exclusively by methane-producing archaea called methanogens. These organisms possess several unique cofactors, such as coenzyme F420 and methanofuran. Most hyperthermophilic archaea are anaerobic, relying on either chemoorganotrophic or chemolithotrophic metabolism, energy derived from chemical reactions involving organic or inorganic compounds. Sulfur compounds often play a key role in these metabolic processes, where sulfur is reduced, releasing energy. However, an interesting exception is Acidianus ambivalens, formerly Desulfurolibus ambivalens. Order sulfolobles, which oxidizes sulfur aerobically despite its extreme thermo- and acidophilic nature. Most halophilic archaea are aerobic chemoorganotrophs, deriving energy from organic chemical reactions. However, in anoxic conditions or when nutrients are scarce, many extreme halophiles can utilize light energy. They contain bacteriorhodopsin, a protein that absorbs light and catalyzes proton transfer across the cytoplasmic membrane. This generates an electrochemical gradient, which in turn powers ADP synthesis via ATPase. Like bacteria, archaea exhibit extreme morphological diversity. Archaeal cells range in size from 0.4 m, Nanoarchaeum equidens, to 100 m, Methanospirillum hungatii, with an average size of around 1 m. Their cell shapes vary and include cocci, Methanococcus janaskii, rod-shaped forms, Thermoproteus neutrophilus, spirals, Methanospirillum hungatii, lobed cocci, Archaeoglobus fulgidus, discs, Thermodiscus maritimus, long filaments, Thermophilum pendens, and even square-shaped cells, Haloquadratum walsbii. Archaea often possess flagella for locomotion or filamentous appendages, pili, for attachment to surfaces. Most known archaea are extremophiles, meaning they are adapted to the extreme conditions of their habitats. Many species have the ability to thrive at very high temperatures, hyperthermophiles above 80 degrees Celsius, very low or very high pH values, acidophiles and alkalophiles. High salt concentrations, halophiles, or high pressure, barophiles. The hyperthermophilic archaea are commonly found in volcanic environments, both marine, black smokers, and terrestrial, geysers, solfataric fields, such as the volcanic habitats of Yellowstone National Park. Halophilic archaea thrive in high salinity environments, like the Dead Sea or naturally occurring marine brines. Methanogenic archaea are also extreme in a way. They grow exclusively under anoxic conditions, often requiring molecular hydrogen for their metabolism. These archaeal species are relatively widespread, found in freshwater, marine environments, and soil, as well as symbionts in the digestive tracts of animals and humans. Archaea have even been detected in human naval folds, though such occurrences are rare. Due to their extremophilic nature, archaea were initially considered to have limited ecological significance. However, recent advances in molecular biology detection methods have revealed that archaea exist in large numbers even in relatively cold seawater, as well as in soil and freshwater habitats. In some oceanic regions, Crenarchaeota can make up 90% of the existing organisms. Overall, estimates suggest that Earth's oceans contain approximately 1.3 times 10 superscript 2 archaea and 3.1 times 10 superscript 2 bacteria. Despite these findings, most archaeal species that have been isolated and cultivated in pure laboratory cultures remain extremophilic, though some have been successfully grown under less extreme conditions. Research suggests that archaea play a significant role in nitrogen, carbon, and sulfur cycles within Earth's ecosystems. The classification of archaea as a distinct domain is based on a set of genetic, physiological, structural, and biochemical differences, particularly marked differences in the sequence of ribosomal RNA, specifically 16S rRNA. In the late 1970s, American microbiologists Carl Woese and George Fox identified archaea as an independent systematic unit alongside bacteria, eubacteria, and eukaryotes. Their research revealed significant differences in ribosomal RNA sequences compared to bacteria, as well as unique cellular structures and metabolic traits that pointed to a separate group of prokaryotes. These findings were later confirmed, leading to a major taxonomic revision. Eubacteria were renamed bacteria, and archaebacteria became archaea. In 1990, Archaea and bacteria were recognized as two distinct domains, alongside Eukarya. In the three-domain system, phylogenetically, Archaea are likely more closely related to eukaryotes than to bacteria. While Archaea lack membrane-bound organelles, they can form filaments resembling a cytoskeleton to help stabilize their shape. There is a defined procedure for describing genus and species. Through publication or validation in the International Journal of Systematic and Evolutionary Microbiology, IJSEM. The genus and species are officially recognized. Higher taxonomic ranks can also be described in this journal. The current status of taxonomic classifications can be found in the list of prokaryotic names with standing in nomenclature, LPSN, maintained by Jean Usaby. This follows the regulations of the International Code of Nomenclature of Prokaryotes, ICNP. Taxa that do not conform to this standard are denoted with quotation marks. Additionally, the global classification of Archaea and bacteria has been reformed through phylogenetic analysis of the 16S rRNA gene. A comprehensive collection of taxa from this and other related publications is available in Berge's Manual and the Taxonomic Outline of Bacteria and Archaea. Over time, additional phylogenetic marker genes have been incorporated alongside the 16S rRNA gene. Some taxa, while legitimate, have not yet been validly published or are not officially recognized by the ICNP. Such taxa are also presented in quotation marks. The classification presented here includes taxa from phylum to family. In some cases, conflicting entries exist, and these have been evaluated based on original literature and phylogenetic analyses for accuracy. A few years ago, two additional phyla, Coarchaeota and Nanoarchaeota, were described. A representative of the proposed phylum Nanoarchaeota, Nanoarchaeum equitans, was successfully co-cultivated and its genome was sequenced. The proposed Coarchaeota phylum was initially identified through 16S rRNA gene sequence analysis in samples from hot springs, and enrichment cultures have since been developed. The complete genome sequence has now been published under the informal name Candidatus Coarchaeum cryptophyllum. However, without isolated strains, representatives of these phyla do not have a validated place in taxonomy under current ICSB regulations, yet they still represent two of the four known archaeal phyla. The following article explores a fictional evolutionary history in which originally unicellular archaea gave rise to a highly specialized, socially organized system, a process unique in its kind. This lineage, represented by the Pokemon Mook, illustrates how microscopic organisms in extreme habitats can collaborate to develop a state-like structure similar to an ant colony, specifically in muddy environments. As part of this reconstruction, the new order Tellurales and the family Ludicolaceae are assigned a place within the fictional tree of life. In an alternative scenario of life's history, Archaea developed structures far beyond traditional individual survival. A gradual evolutionary specialization led to the formation of complex colonies, where individual cells became functionally differentiated and collectively shared tasks. This represents a transition from originally isolated extremophilic microorganisms, collaborative organisms emerged, forming an organized social collective, much like ant colonies and mud nests. The lifestyle and physiological traits of these organisms offer insight into how evolutionary strategies can foster multicellularity and division of labor in modified, unconventional habitats. The name Tellurales derives from the Latin tellus, the earth goddess, and the term for earth, symbolizing the terrestrial origin of these organisms. Similarly, Ludicolaceae combines the Latin root ludum, mud, clay, with the suffix colaceae, commonly used in family names, emphasizing the strong connection to the muddy habitat where the colony originates and thrives. This classification is, of course, a constructed concept, based on the premise that social interactions and specialized division of labor had already emerged as survival advantages in the earliest evolutionary phases of Archaea. Members of Ludicolaceae exhibit remarkable cellular architecture. Instead of rigid cell walls, their flexible membranes are enriched with unique lipid components, characteristic of Archaea. This enhances their resilience and adaptability to changing environments. A key feature is the production of exopolysaccharides, forming a sticky, slimy matrix. This matrix not only serves as a physical connection between cells but also acts as a nutrient reservoir and a medium for chemical signaling. Already at the cellular level, a proto-multicellular work process is evident. Some cells specialize in reproduction, analogous to queen insects, while others function as sterile worker units, responsible for nutrient accumulation, colony maintenance, and defense mechanisms. These traits allow the colony to behave as an integrated organism, a phenomenon not observed in real microbial archaea, but plausible within this fictional scenario. The habitat of these organisms is primarily muddy, moist substrates, where nutrients are unevenly distributed and scarce. The Ludicolaceae represent a unique ecological strategy. In densely populated biofilms, a network of interconnected cells forms a state-like organization. Through chemical signals, comparable to pheromones in social insects, they coordinate movement, nutrient retrieval, and defense against competitors. The slime matrix acts as a protective barrier against physical threats and harmful chemicals, while also creating microhabitats that stabilize temperature, pH, and humidity, essential in harsh environments. In response to environmental changes, the colony demonstrates rapid adaptability. Within the slimy network, cells can shift into specific functional roles based on need. On a small scale, this mirrors task division in ant colonies, where each caste contributes to collective survival. The emergence of collective lifestyles in Ludicolaceae represents an evolutionary leap in innovation. Originally, unicellular archaea that existed in isolation within extreme niches likely made their first attempts to form small but stable groups. Environmental stress, such as limited resources and high competition, promoted selection for individuals that benefited from communal living. Horizontal gene transfer and targeted mutations contributed to the development of specialized signal transduction mechanisms, which drove differentiation within the colony. This enabled individual cells to release signaling molecules, triggering the reprogramming of neighboring cells, a precursor to more complex sociocellular regulation systems. The parallels to social insect societies emphasize how similar evolutionary challenges, such as survival and variable resource-scarce habitats, can lead to comparable solution strategies, even across domains. The Ludicolaceae thus represent a case of convergent evolution, where microorganisms cultivate collective intelligence and self-organization to ensure their survival. While classical archaea typically function as single, independent cells, the Ludicolaceae model offers new insights, like ants, which rely on sophisticated communication and role distribution to form complex colonies. The cells of Slimocaceae exhibit remarkably organized behavior. Although their forms are completely different, the principles of resource distribution, protection, and collective adaptation are strikingly similar. The aggregation of unicellular organisms into a multicellular structure is also observed in slime molds, further illustrating how cooperation among small units leads to emergent properties that open new ecological niches for the collective. These comparative perspectives expand our understanding of how different life forms, from bacteria and archaea to insects and fungi, address similar evolutionary challenges. That's it for now. In the next episode, we'll take a closer look at Cloyster and its relationship with all insect Pokémon. Feel free to subscribe to our channel, leave a like, and check in next time.